new concept, a new way to try to treat this disease, which really had a huge impact on our society. We'll talk about that. So cancer is, thanks very much, cancer is a big problem, of course, we all know. And one of the problems with cancer is that we hear about it as a single word, cancer. In fact, it's many, many different diseases. It's different as you and I and everybody in this room. Each person's cancer is very unique because it's a change in the genetic information. I'll talk about it more in a second. And clearly, devastating society. This number is 72,000 Canadians will die of this disease. Of course, the impact of society is much bigger than that. Of course, the families of people who die of disease are also affected by that, but also people who are treated with disease. Many people never go back to work. No longer are functional members of society, so it's a real problem being sent out. Around the world, it's going to be an absolute disaster. I was in China last year, and the numbers of people in China who are suffering from cancer and really have essentially no treatment is staggering. So it's a worldwide problem. Part of the problem we have is that unfortunately our treatments are not as good as they need to be. So this is a picture here of a woman undergoing therapy for breast cancer, and you can see that it's an image that many of you will be familiar with. The unfortunate part is therapy is attacking their cancer, which is good. Probably a good outcome. But unfortunately, it's also attacking some of her normal tissues. So she's lost her hair. She's probably had her immune system suppressed. The work that Susan Dent's doing about cardiac challenges with patients undergoing cancer therapy is another great example. The problem is these therapies, although they're effective, attack both the normal tissues and the tumor. And it's very hard to sort of tweak them, get them just right, so you cure the patient without causing some damage. And so my lab and many labs around the world are trying to change that. We're trying to come up with what we call cancer-targeted therapies. And these are therapies that hit the bullseye, hit the tumor, and not hit your normal tissues. So it seems like an easy thing, or it's a good idea, of course, but it's not such an easy thing to do because, as I'll talk about in a minute, there are just a few seconds about the biology of this disease. It's really hard for drugs to distinguish between cancer cells and normal cells. And that's really been a challenge in my lab and thousands of labs around the world working on this problem, trying to come up with new cancer-specific targeted therapeutics. So I'm going to tell you sort of how I got into this problem, how I understand, the way I look at it, and how I come up with some solutions. And the important thing, I think, is to think about, from my perspective, is to think about genetics. And I think it's fair to say that genes do control all aspects of our growth. We have genetic information that tells us to become who we are. And just as an example, this is a picture here of a human egg that's been fertilized. And when I went to university, I thought this is an amazing story. That human egg, which we can only see with a microscope, has all the information in it to make another human being, which is phenomenal. And after a few hours, we now look like this. We're now embryos, which have multiple cells in that make up the person. So now you have, those are all individual cells. And each of the cells in that embryo, again, have all the genetic information to make another human being. It's phenomenal. But over a few weeks, we now look like this. And so when I think about it, we go from that single cell to this person who's made up of billions of cells in a matter of weeks. It's an incredible biological phenomenon, great orchestration of genetics, and it's just fantastic. And then over time, over a few decades, things don't necessarily turn out like we like. And Homer himself is a combination of his genetics. So he's lost his hair probably because of his genes, but he's also got this thing going on here, which is because of his environment. We now know that cancer is a disease that's caused by interactions with our environment, and it's affecting our genetic information. So what do I mean by that exactly? Well, we're all made of billions of cells. So let's pretend that's your skin there, and that little blue rectangle is the cell that makes up your skin, and the oval is your genetic information. So we're made up of billions of these cells. Our skin replaces itself every single day. We make a billion blood cells every day. It's a phenomenal thing that we do. It's all genetic control. There are genes that say, hey, it's time to duplicate yourself and make another sister cell, and that's controlled by growth genes that say it's time to make a copy of yourself. You see that cell is divided and made a duplicate of its genetic information. Oops, Paramount's trying to re-advertise me here. Okay, that's genetically controlled. What we now know from years of studying biology of humans and mammals is that there's also genes inside our bodies that say it's time for you to die. There are so-called cell death genes that say to every cell, hey, you live long enough, it's time for you to die, go away, So what happens in cancer? In cancer, you know, we do things we shouldn't do. We smoke when we shouldn't. We go to tanning parlors. We live in big cities like Ottawa where there's pollution, traffic, and so on. Steel plants in Hamilton where I grew up. But also, some agriculturists that, like, if you think about it, if you have to completely replicate genetic information 1 billion times every day, the chances that your cells are going to do that perfectly every time is probably pretty limited. So you're going to make mistakes in the copy of genetic information. And so all these things contribute to the events which I would call mutations, which can happen in individual cells. And if by chance the mutation occurs in one of your genes that happens to be a cell death gene, that red cell now has all the genes it needs to grow, except it's lost the cell death genes. And so it continues to grow and grow because it's got all the right things to die. But it never knows how to die. It becomes immortalized. And we now know from the last 20, 30 years of research in cancer that this is an important contributor to cancer. This genetic changes in cell death genes allow cells to become immortalized, grow forever, and form tumors. I have cells that I'm growing in my lab. The wife's name from a woman in 1953 who had cervical cancer. She's long passed away, but her cells continue to grow in my lab because they've been immortalized by these sorts of mutations in cell death genes. Okay, so what does this all mean? This is all biology. Just to reiterate, normal cells, because of mutations in individual genes, become tumor cells. And what we discovered is that the same genes that control cell growth and death, in other words, those genes that must be mutated where normal cells become a tumor cell, turns out those same genes control the ability of our individual cells to fight virus infections. So that means that when a tumor cell becomes a tumor cell, it throws away some of these cell control genes. It also throws away the ability of that tumor cell to fight virus infections. Okay? That makes it a target. We now have a target in that tumor cell that we can potentially attack. So what do I mean by that? So just to tell you a little bit, almost finished biology here. We're made up of billions of these big round cells here without genetic information inside them. That's what makes up our body. If a cell is that big, then a virus would be this big. It'd be extremely small, hard to see. We actually only visualize an electron beam. You can't even see them with light. And genetically very simple. Viruses cannot exist on their own. They're true parasites. They have to move inside the cell and use the cell's genetic information to make copies of themselves. And then they can move on to another cell. It turns out that viruses can do this, and when they do this, they actually kill the cell that they infect and destroy it. There's lots of viruses out there that we get infected with viruses and so on, that often we're able to eliminate, but in the short term they infect our normal cells and destroy them. Our idea is we actually make viruses specifically and only parasites of tumor cells. And we have such things. This would be the target therapy we want. We can infect the tumor and destroy it. It can't infect your normal tissues. You don't get any of these off-target side effects. Maybe that's what we're looking for. So we did create some viruses that are now tested. I'm going to tell you a little bit about these. So these are viruses that can specifically infect and kill tumors, but cannot infect your normal tissue. So if we were to squirt these viruses inside you, and you didn't have a tumor, those viruses would swim around, they'd have nowhere to grow, and they'd eventually be eliminated. So the one I'm going to talk about first is this one here. It's called vaccinia virus. And most of the people in this room probably have a scar on their shoulder, because you were vaccinated with this when you were younger to prevent you from getting smallpox. This is not smallpox, but it looks enough like smallpox to your immune system that you get vaccinated with this, it protects you from smallpox infection. The reason I'm telling you that is because this virus has been given to really millions and millions of us around the planet as a vaccine, and it's perfectly safe. So we know that it's not very good at infecting normal tissues, so that's when we started trying to make something that would be a parasite of tumor cells. And when we created this thing, we needed to have a test bed to see how we expect to see it work. And so we use mouse models all the time. So we have mice in my lab, which will inject, for instance, into the tail vein, a colon cancer cell, and that cell will fall over the mouse and eventually see the lungs, and it makes those mice very sick. In fact, if you look at the lungs, this is what lungs of the normal mouse look like. There's the heart there, it's the normal looking lungs. And after we give them cancer, the lungs look quite horrid. Oops. Lungs look quite horrid, they're full of tumors, they're massively worried about tumors now. We say, okay, can we test our idea now? Can we give them a virus that's supposed to be only going to tumor cells, let it go everywhere in the mouse's body, see if you can find all those white nodules in the lungs, infect them, and destroy them, and not affect the mouse's normal tissues. And so just to show you this experiment a little more clearly here, normal lungs, full of tumors, we now give into the blood vessels of those mice, so intravenously, we give them a virus. And this virus, we've engineered it to express a protein, which is, you know how in jellyfish, they glow in the water? We take the gene out of those jellyfish and put it inside our viruses. So now wherever the virus infects a cell and uses the cell to make copies of the cell, that cell will turn green. So if we do this now, we look 24 hours later, what do we see? We see this amazing picture, which is all those white lumps are turning green because they've been infected by the virus. But if you look in this region right about here, where there's normal lung tissue, and look down here, you see it's black. So this virus is doing exactly what we want. It's infecting tumors and not normal tissue. It potentially is a target therapeutic we can use to treat people. And so we did this experiment now in people, and we're beginning to see some interesting results that we're excited about. So these mouse experiments sometimes don't pan out, but sometimes they do lead to important therapeutics. So here's a gentleman who had liver cancer, and unfortunately, during this disease, he got this large tumor on his neck from his liver. And it got to be so big that he couldn't turn his head anymore. He was suffering from this disease. He was losing weight. He was on his way out. It was not looking very good for him. We were allowed to inject him with one of our viruses. And as you can see, that tumor eventually completely resolved. He began to gain weight again and actually became healthy again. It's really a very nice response. This gentleman here is a real superstar in our trial. And so on the top, I want you to look, those are two CT scans where we're looking down from someone's body in this plane. If you look over here, that thing inside the red circle there is a kidney tumor that's now metastasized into his abdomen. So just to give you some bearings, that would be his spinal column there coming up towards you. And after you see treatment with the virus, you see that tumor's gone. Oops. How am I doing this? There you go. Yeah. You can see that tumor's gone. So quite remarkable. And a whole body scan, which is on the bottom there, you can see these circles show where the tumor was before and after treatment it's gone. His whole body scan. So really quite a remarkable response. This gentleman has gone back to work as a construction worker. So really profoundly changed someone's life when this starts to work correctly. This is a lady in Hamilton that we treated. You're looking at where's the baseline over here. You're looking at her liver and this ugly thing hanging off the end is a tumor. It was treated, and after a few days it started to change shape, change the way it looks. Eventually it completely is eliminated, and she's about